Welcome back after the post-lunch session. Post-lunch will start with a symposium on ANCA-associated renal disease. To moderate this session and introduce the speakers, we kindly request Dr. Swarnalata Madam, HOD of NIMS, Hyderabad. Thank you.
So good afternoon. It's been a great going. We should congratulate the organizers for a wonderful scientific program. Though I see a less number of participants here post-lunch, I hope people will join once we start the program. So let us now move on to the next interesting topic, that is the ANCA vasculitis associated kidney diseases. It's been a heterogeneous autoimmune diseases with a wide variety of presentation, and though it's rare, it's been devastating, resulting in either the patient mortality or the organ failure. There has been a lot of change in the understanding of pathophysiology and the treatment, and particularly with respect to the plaques and then the newer molecules. So let us now listen from the young nephrologists across all the southern states. So we have five speakers, and the topics have been divided accordingly. So I'll just try to call one after the others. They can come onto the stage and they can join me. And after we have all the talks over, we'll have 12 minutes time for each of the talk. So after finishing all the 5 talks, then we'll invite the comments from the audience. So let me first invite Dr. Saivani. She is a young nephrologist working in the Kannur Medical College from Andhra Pradesh. I would invite her onto the stage and request her to talk on the pathogenesis of the ANCA vasculitis and glomerulonephritis. Over to you, Dr. Saivani.
Good afternoon, everyone. At the outset, I would like to thank the organizers for giving me the opportunity to be part of this wonderful program. And my topic of this symposium is Pathogenesis of ANCA vasculitis and ANCA-related renal disease. So, Chappell classified the vasculitis into large vessel vasculitis, medium vessel vasculitis and small vessel vasculitis. Coming to small vessel, there are 2, immune complex deposition and no immune complex deposition, that is nothing but quasi-immune, and that is now named as ANCA vasculitis. Coming to ANCA vasculitis, it is a life-threatening group of multi-system diseases characterized by small vessel quasi-immune vasculitis, presence of circulating pathogenic ANCA with absence or paucity of immunoglobulin deposition in injured vessels. ANCA-induced activation of prime neutrophils and monocytes leading to destructive vascular necrosis. Here, neutrophils plays a central role as both effector cells and targets of autoimmunity. So, what is ANCA? It is predominantly immunoglobulin G autoantibodies directed against neutrophil primary granules and also lysosomes of monocytes. There are 2 assays, indirect immunofluorescence assay and enzyme immunoassay. We should do both the tests because it increases the sensitivity. According to immunofluorescent pattern, they are classified as cytoplasmic ANCA and perinuclear ANCA and X-ANCA with variable fluorescent pattern, and enzyme immunoassay anti-MPO, anti-PR3 and LAM2 antibodies. It has different roles as serological diagnostic markers and pathogenic role. The titers are unreliable indicators of disease activity. Depending on titers, we should not go with the treatment. Coming to history, in 1954, Goodman and Shirk found out there were similarities between granulomatous polyangiitis, eosinophilic granulomatous polyangiitis and microscopic polyangiitis. In 1979, a good study by Stillman et al., they have seen 46 cases of crescentic GN. Among them, there were no immune complexes in majority of the cases. In those cases, 1988, Falk and Jennett find that ANCA were present in circulating in the blood. 85 to 95% of those cases. So in 2012, Chapel Hill proposed nomenclature of ANCA vasculitis, and they have renamed all Wegener's, Church's, Truss. And in ANCA vasculitis, it involves the small vessels like capillaries, venules, arterioles and small arteries. In microscopic polyangiitis, it causes necrotizing vasculitis but without any granulomas. In granulomatous polyangiitis, there is the presence of necrotizing granulomatous inflammation. And in eosinophilic granulomatosis with polyangiitis, there is increased eosinophilic deposition in the granulomas. And this slide depicts the incidence and presence of PR3 ANCA, MPO ANCA, and rate of renal involvement is more with the microscopic polyangiitis, and majority of the presentation is like RPGN. So, this is a study done in University of North Carolina Nephropathology Laboratory. In 232 renal biopsy specimen, the diffuse necrotizing and crescentic glomerulonephritis tops the list. That is nothing but these people mostly present like a RPRF. Coming to pathogenesis of ANCA, it is a conglomeration of genetic, environmental and infectious things. And coming to genetic, because striking white predominance suggests a genetic role, but there is no familiarity. Suggests that apart from hereditary factors, some other factors also may play a role. Coming to genome-wide association studies, there are MHC and non-MHC gene loci which provoke more GPA and MPO ANCA-associated vasculitis. These are all the HLA genes which are responsible for this ANCA vasculitis. Coming to environmental exposures, smoking, exposure to silica dust, solvents and drugs, they predispose the ANCA vasculitis. Coming to infection, there are entamoeba, histolytica, staph aureus. There are many studies which show the presence of these infections will predispose the ANCA vasculitis. The clinical evidence supporting the pathogenicity of ANCA, whether these ANCA are present silently or these are the main causes for pathogenicity. Coming to human models, a neonate presenting with pulmonary hemorrhage because of transfer of ANCA antibodies from her mother. And another, vascular inflammation is highly correlated with the presence of ANCA in the circulation of these patients. And presence of ANCA target antigens PR3, MPO and lactoferrin in the fibrinoid necrosis deposits. And also high levels of ANCA are present in active disease and less in remission. In vitro evidence is that when neutrophils are exposed to ANCA in the presence of inflammatory milieu, they cause activation. And in these animal models of mice, when they are exposed to anti-MPO immunoglobulins, they cause crescentic glomerulonephritis. Coming to emergence of ANCA, there is a autoantigen complementarity. That is, adaptive immune response against a complementary peptide. That is, if the patient PR3 gene is there, if complementary peptide, DNA and peptides are produced against that, and this complementary peptide initiate a idiotypic response and produce a majority of ANCA, and they react with the PR3. Coming to molecular mimicry, it comes from sequence homology between FIMH, which is a gram-negative bacillus adhesion molecule, and there is a mimicry between lysosomal membrane associated protein 2, LAMP2. So this is the diagram depicting the role of complementary peptide, that is, transcription of endogenous antisense strand of DNA will produce these complementary peptides. These complementary peptides activates the B cell and turns into a type 2 phenotype and produce the majority of the ANCAs and which activate the neutrophils. And here these are the FIMH which are having molecular mimicry with the LAMP2, and they activate the B cells and produce the ANCA. Coming to toll-like receptor pathways, the ability of staph aureus to activate neutrophils through toll-like receptors. So they think toll-like receptors also are having importance in pathogenesis. And drug-induced ANCA like hydrologin, propylthiouracil and levamisole adulterated cocaine have been reported to be strongly associated with the ANCA vasculitis. Coming to how this ANCA produce the activation of neutrophils and monocytes. First is the priming. In the presence of inflammatory milieu like TNF-alpha, LPS and interleukin 18, the ANCA causes priming of ANCA antigens onto the surface of neutrophils. That is MPO and PR3 comes onto the surface of neutrophils. That is called priming. And this PR3 membrane expression, if it is high, it increases the activity, and also it is high expression is seen in the relapsed patients. And this is CD177 and PCAM1 platelet endothelial adhesion molecule 1 interaction, which cause neutrophil and endothelial transmigration and which increases the vascular damage, because vascular damage is the ultimate goal of ANCA. Coming to Fc gamma receptor binding, the binding of ANCA with Fc gamma receptor perpetuates the neutrophil activation. Here, when neutrophil and monocytes are activated by Fc gamma receptor, neutrophils produces serine proteases. These serine proteases are mediated by cathepsin, and all these will produce the interleukin 1 beta, which is the most pathogenic cytokine. And these are the target things we can use it for the treatment. NOX2 inhibits the cathepsin, so interleukin beta can be decreased, and anti-interleukin beta drugs can be used to treat the ANCA vasculitis. And neutrophils and endothelium interaction by beta-2 integrins and ICAM1 will increase the further expression of PR3 on the neutrophils, and also endothelial activation and diapedesis of the neutrophils. And expression of PR3 on the neutrophils on the apoptotic cells is the main trigger, and which causes phagocytosis of PR3 cells by macrophages, and they produce the chemokines and they recruit the inflammatory cells. Coming to netosis, now these nets are coming to limelight in both ANCA and also SLE. It was first described in 2004, and they thought that it was having antibacterial role. And it consists of bacteria trapping and are composed of decompensed fibers of chromatin decorated with the antimicrobial peptides such as neutrophil elastase, MPO, PR3 and others. Excessive netosis is however deleterious, and it has been found in the ANCA associated vasculitis. The importance of nets are, they trigger the adaptive immunity through exposure of extracellular autoantigens by antigen presenting cells, and MPO ANCA have a high ability to generate the nets in vitro and confirm their impaired degradation in ANCA vasculitis patients through weaker DNase activity, and nets could also provide a new link between innate immunity and stimulate the platelet adhesion and coagulation. Finally, nets induce the endothelial cell death through histone dependent cytotoxicity, which is major in the ANCA associated vasculitis. So, this is the diagram depicting where first neutrophils are primed, these small dots are come onto the surface in the presence of this inflammatory milieu, and after activation of these neutrophils and the ANCA present in the blood, they bind with these receptors and they cause the degradation of neutrophils, and the neutrophils produce reactive oxygen species and NSPs, and these NSPs produce interleukins, and neutrophils also damage and produce these nets, and these nets will contain all these autoantigens, and these antigens again initiates the antigen presenting cells and adaptive immunity. And the key role is the loss of self-tolerance. The impaired clearance of apoptotic neutrophils and lower level of or dysfunctional B and T regulatory lymphocytes is the major thing which continues the cycle. Survival of autoantibody secreting B cells is dependent upon additional factors like BAF and B-Lys, and these B cells are very important because activated B cells are present in the granulomas, and rituximab is the treatment of choice. So, B cells are very important. Coming to B-Regs, B regulatory cells will increase T regulatory cells, and they also enhance the apoptosis of T cells. And coming to T cells, T cell activity is increased in and they are present in the lesions, and there is interleukin 2 activity is high in these ANCA vasculitis patients, and there is apparent T helper polarization. Th2 phenotype is seen in generalized ANCA vasculitis, Th1 in localized, and Th17 which comes recently in the limelight. They are causation are important in inflammatory milieu causation and neutrophils and macrophages deposition. Coming to T effector memory cells, they are important biomarkers and their downregulation is important. And T regulatory cells, their qualitative disturbance, because FOXP3 is having loss of exon in the ANCA vasculitis. So, not the number and the quality is important in T regulatory cells. Coming to neutrophils, they are important of source of TNF ligand forming inflammatory milieu, and also they secrete BAF, Baileys, April. These all things are important for maintenance of B cell differentiation and production of ANCA. So, this is the important diagram. Here effector memory cell and Treg cell disbalance increases the problem, and aberrant T helper cell polarization 1, 2 and 17, and Breg cells are inhibiting this aberrant T cell polarization and also activating Treg cells and also inhibiting T helper cells to activate the B cells, and these neutrophils secrete BAF, Baileys and in turn activate the B cells, and after that neutrophils activating producing NETs and interacting with the endothelium. Alternate complement pathway is also came into limelight. Initially they thought complement has no role, but C5, BB in all mouse models, alternate complement deficient mice have protective response, and C5AR deficient and C5AR antagonist treated mouse are protective against ANCA vasculitis, and factor BB levels correlate with crescents, and complement factor H inversely correlates with the disease activity. These can be taken as biomarkers, and all these show that importance of C5AR antagonist and C5 inhibitor eculizumab can be used as a treatment for ANCA vasculitis.
Can you please conclude?
Yes, ma'am. So, this is the diagram showing importance of complement. And these are the various treatment targets which can be dealt with my subsequent speaker. And coming to take home message. So, ANCA vasculitis involves immune and non-immune factors, and it involves activation of prime neutrophils damaging the endothelium, and neutrophils play a central role in the pathophysiology, and the NETs and alternate complement pathway activation are the emerging concepts, and adaptive immune cells are activated due to insufficient regulation by Treg and Breg cells. Thank you for giving me this opportunity.
Thank you, Saivani. You have beautifully, concised all pathological mechanisms of the ANCA vasculitis and renal diseases. So we'll take up the questions at the end. You can just please join me.
Okay,
so actually, you can just join me here.